Welcome back, folks. Still thinking about the lipids in membranes, the fluid part of membranes. So still thinking about that. And what I want to look at now, though, is some inherent asymmetry that you find in biological membranes, and this applies to the plasma membrane, cell membrane, and also membranes that are internal. So the membranes around compartments in the cell. So membrane asymmetry refers to the difference between the monolayers or the leaflets regarding the kinds of lipids that are there, and then also the degree of saturation of the fatty acids in those lipids. So there are kind of two things going on there: the length of the fatty acid chains, but also the type of head group that we find there. So there are those two things that contribute to the diversity that gives us the asymmetry. So let's just kind of look at things generally. If we look at total membrane lipid on the y-axis here, and then we've got these different categories of types of lipid: sphingomyelin, phosphatidylcholine, phosphatidylserine, phosphatidylethanolamine, phosphatidylainositol. Is what we see is that there's this huge difference in the types of phospholipids we find in the different leaflets. So this is the outer leaflet, and so you can see it's enriched for sphingomyelin, phosphatidylcholine. But then if we look at the inner leaflet, we find that it's enriched for phosphatidylserine, phosphatidylethanolamine, and phosphatidylainositol. So what is the reason for this? Or why do we find this membrane asymmetry? Why do we find a different set of phospholipids in the outer membrane leaflet, or the outer leaflet of the membrane, versus in the inner leaflet? So before we get to that, let's just talk about how we might get that asymmetry. So once you establish this asymmetry, it only changes under certain physiological conditions in response to certain cellular events, if you like. You obviously know that membranes can move laterally and rotate in the membrane, but that they can't readily flip from one side to another. So once you establish this asymmetry in the membrane, you're kind of stuck with it unless you have a mechanism for moving lipids backwards and forwards across the membrane. Now that's energetically unfavorable, and so it's only going to be done if there is a physiological need for it, and so we'll look at those in a moment and have a couple of examples. That's the kind of setup for the diversity. Let's just kind of, or the asymmetry, let's just look at some of these in more detail. Glycolipids tend to be found on the outer leaflet of the membrane, and then we've got a lot of phosphatidylcholine and sphingomyelin on the outer leaflet. So we don't tend to find many glycolipids on the inside. So in a moment we'll take a look at why that might be. On the inside, we'll take a look at why we have this abundance of phosphatidylainositol and this abundance of phosphatidylserine. Phosphatidylethanolamine, we've already talked about a little bit. That's what gives us curvature. So if we want to curve that membrane, then one of the ways we can do that is by having a particular distribution of phosphatidylethanolamine. So let's kind of take a look at some of the reasons why we might have some of this diversity, and some of them will be a little familiar to you. Let's start off with the glycolipids. Now, glycolipids, and for that matter glycoproteins, they are found predominantly in the outer leaflet. So if you have a protein which has been glycosylated in the ER and the Golgi, it's going to generally be in the outer leaflet, and you can think back to why that might be based on how proteins are glycosylated, and we'll come to that in more detail later in the semester. So here's a nice example that you'll be familiar with about why we have asymmetry in regard to glycolipids. The example here is the ABO blood group system. So the ABO blood group system arises because this oligosaccharide here of glucose, galactose, N-acetylglucosamine, and fucose, all of these sugars are added to lipid or protein, and when that happens, you get A antigen. Now, A antigen is derived from O antigen. So if you remember general biology, when we talk about O antigen, we say there are no sugars on the cell surface. But it is not entirely true. There are some sugars on the red blood cell surfaces when you are blood group O. It's just that it's the base sugar. So at that point, it's easier for us to say there's no sugar there at all. So there's this base sugar, and then we can either add on N-acetylgalactosamine here to make A antigen, or we can take the O antigen carbohydrate and we can add galactose to make the B antigen. So depending on what combination of these sugars you have on your lipids or proteins in the outer leaflet of your membrane, you'll have A, B, or O blood. And so if you have type A blood, you're going to have some of this A antigen. If you have B type blood, you're going to have some of this B antigen. O, you're going to have this sugar. And so if you're AB, you have a mixture of this sugar with this N-acetyl-galactosamine added, and some of this sugar with galactose added to the base O antigen sugar. So depending on whether you're AB, A or B or O blood group, you are producing a set of enzymes in your cells which basically work at the level of the Golgi in the ER to build these different sugars and add them onto lipids or proteins, which then end up in the cytoplasmic, in the exoplasmic leaflet, the outer leaflet of the plasma membrane. So that's one of the reasons why we have asymmetry in membranes. That's an important part of the cell signaling to its environment something about the characteristic of the cell. It says we're type O blood cells, it says we're type A blood cells, and so on. So that's like a neat example about why you have membrane asymmetry. It'd be no good if this was on the inside of the membrane, because then the immune system of the body can't tell what the blood type is, and so you must have this on the inside, on the outside leaflet. Another couple of examples we can just do very, very quickly. I initially said that once you establish this asymmetry, which is established during membrane biogenesis, when you establish this inherent asymmetry, you can't really change it. You can, but only in response to certain physiological responses. So one of those times when you actually do change the abundance of the phospholipids in the different leaflets is when you have to have blood clotting. So one of the things to stimulate blood clotting is that in a platelet cell, the intracellular calcium goes up, and that leads to an activation of an enzyme in the membrane called scramblase. So this is a transmembrane protein called scramblase, and what that does is it takes phosphatidylserine and it flips it from the inner leaflet, where it's more abundant, to the outer leaflet, where it's less abundant. So for a cell which is flipping its phosphatidylserine, one of the things for platelets that signals is we need to stimulate blood coagulation, and what that does is the phosphatidylserine forms these rafts which recruit these blood factors XA and VA, and they convert prothrombin to thrombin, and that drives blood coagulation. So what's happening here is that we're keeping the phosphatidylserine predominantly in the inner leaflet, but hey, when we need a clot of blood, the cell is going to flip the phosphatidylserine, and that's going to act as a mechanism to stimulate blood coagulation. Another really kind of common and interesting example of looking at lipid asymmetry changing is in triggering the apoptotic response. So apoptosis we'll come to later, but generally it's a mechanism which cells engage when they need to trigger their own death. So it's a controlled form of cell death, and that's under the control of a whole bunch of proteins in the cell called caspases. And caspases can do one of two things to some proteins at the membranes. First of all, they inhibit an enzyme which brings phosphatidylserine from the outer leaflet to the inner leaflet, and so this enzyme is maintaining this asymmetry where the phosphatidylserine is in the inside leaflet of the cell membrane, and the caspases activate this enzyme scramblase. And so what scramblase does is it starts to flip the phosphatidylserine. So the phosphatidylserine gets flipped out, and then other cells like macrophages can recognize this cell as undergoing apoptosis, and this cell will be starting to break up. So the macrophage is recruited by the phosphatidylserine and some other things that are going on in the outer leaflet of the membrane, and it can start to clean up the pieces of the cell which start to disintegrate and bleb off, as it's called. So there's some information about membrane asymmetry and the biological relevance. I'll be back soon.